Today we're going to talk about the second half of gene expression, or the second half of the central dogma, translation. For this lecture, make sure that you take plenty of notes, putting the information that you see from the slide in your own words. Before the lecture begins, make sure that you've read the assigned portion of your textbook. As you listen, make sure that you've minimized all distractions, turning off music, TV, movies going on in the background. Listen in a very quiet space so you can give it your full attention. All right, let's get started on translation. Remember that DNA is transcribed to mRNA, which is translated into proteins. DNA can have mutations, RNA undergoes post-transcriptional modifications, and proteins undergo post-translational modifications. Remember also that nucleic acids are what regulate gene expression. DNA encodes the genes, and the DNA is found in the nucleus. mRNA is made from the DNA, and again, this occurs in the nucleus. So transcription occurs in the nucleus, and the mRNA has to leave the nucleus through a nuclear pore in order to make it to the ribosome to make the polypeptide chain. So today we're going to look at translation, the second step in this central dogma. DNA serves as the template, and transcription occurs forming mRNA in the nucleus. Translation occurs, and protein is formed by a polypeptide amino chain, and this occurs in the ribosomes. Translation is the conversion from nucleotides, the language of mRNA, to amino acids, which are the language of proteins. These are the stages of translation: initiation, elongation, and termination. During initiation, mRNA, tRNA, and the ribosome form a complex. During elongation, the ribosome moves from the 5 to the 3' end and synthesizes a polypeptide chain of amino acid. Termination occurs when the ribosome reaches the stop codon and disassembles the complex. The completed polypeptide is then released from the ribosome. Again, by way of overview, transcription is the synthesis of mRNA using DNA. Translation is the synthesis of polypeptides using information from the mRNA. Translation occurs in the ribosomes, and today that's what we're focusing on, is the steps of translation. So let's focus on the individual steps that take the mRNA and create a polypeptide chain. Amino acids are composed of an amine group, a carboxylic acid functional group, and a side chain that's specific to each amino acid. There are hundreds of amino acids, but there are 20 that are common because of their role in protein production. The genetic code is made up of codons. The codons are 3 units of nucleotides. AUG is the start codon, and it's shown in the figure on the right in green. There are 3 stop codons that do not code for an amino acid, but instead terminate translation, and they are shown in the figure on the right in red. tRNAs are adapter molecules that recognize these codons in the mRNA. The tRNAs then recognize the codons and link those codons to a specific amino acid that's coded for in the mRNA. The tRNA contains an amino acid attachment site and an anticodon that recognizes the complementary codon on the mRNA. The ribosome is either in the cytosol or attached to the ER, and this is where translation occurs. The ribosome is the site of translation, and it contains 3 sites within the ribosome subunit. It contains the A site, the P site, and the E site. tRNAs move through these sites in this order. The first step in translation is initiation. The small ribosomal subunit binds to the mRNA. The mRNA then binds to the initiator tRNA that has the start codon. All of this occurs as energy is infused into the system. The large ribosomal subunit completes the initiation complex. So the initiation complex for translation includes both the large and the small ribosomal subunits, mRNA, initiator tRNA, and energy. As translation moves into the elongation phase, this is what occurs for the initiator tRNA. It moves from the acceptor site to the P site, so from the A to the P. Now the acceptor site is empty. The tRNA that matches the anticodon to the codon on the mRNA enters into the A site. This shoves the neighboring tRNA over to the P site. As this happens, the polypeptide that has been started on the very first tRNA transfers over to the amino acid that's located on the tRNA on the A site. Once the amino acid chain from the P site transfers over to the A site, then the tRNA that is on the P site moves to the E site, which is E for exit. And now the tRNA that contains the polypeptide chain moves from the A to the P site. At the very end of this first stage of elongation, you have a tRNA sitting in the P site with a polypeptide chain, and this continues as each codon is read on the mRNA until a stop codon is reached. When the ribosome reaches a stop codon on the mRNA, the stop codon comes into the A site. The A site now has a release factor that recognizes the polypeptide and empties the empty tRNA. This release factor promotes hydrolysis. Once the polypeptide and the tRNA are released, the ribosomal subunits and the other components of the translational complex are disassociated. In bacteria and eukaryotes, multiple ribosomes translate an mRNA at the same time. These are known as polyribosomes. Once a ribosome is far enough past the start codon, another ribosome can attach to the mRNA and begin translation. This is seen in the figure above in a drawing of a polyribosome, or in the figure below with a large ribosome in a bacterial cell, which you can see by TEM. Here is another look at the central dogma. Transcription occurs in the nucleus, and after transcription occurs, post-transcriptional modifications have been made to RNA. The capping and the poly-A tail are necessary for mRNA to be able to exit the nucleus and be stable. The pre-mRNA must be cut and spliced into mature mRNA before leaving the nucleus. From here, the mRNA goes to the ribosome, and this can be a free-floating ribosome or a ribosome associated with the ER. In the ribosome, translation occurs, and post-translational modifications occur after the protein has been made through translation. Let's review the process of translation.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly-A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Now that we've looked at the process of translation, we're going to consider post-translational modifications to proteins. There are several forms of post-translational modifications, including the addition of functional groups, folding, and structural changes. Addition of functional groups can include phosphorylation, acetylation, sumylation, ubiquitination, and methylation, just to name a few. We've covered the central dogma at this point, that DNA is transcribed to RNA, which is translated into protein, and that this is the formation or the foundation of gene expression. So what happens to gene expression when DNA has mutations? First, before we get started on this, what is a mutation? Mutations are changes in DNA structure or sequence. There are several different types of mutations. There are large mutations such as deletions or insertions, and there are small mutations such as point mutations, where there's only a change in a few base pairs. Mutations can occur in somatic cells as well as germline or sex cells. Let's break down the types of mutations. There are silent mutations, where the DNA mutations have no effect on amino acids in the proteins that they encode. These are often found in non-coding regions of DNA, or they are mutations that don't affect the amino acids which are incorporated. There are missense mutations, where the DNA mutation can cause the substitution of one amino acid for another in an encoded protein. Nonsense mutations involve a base substitution that causes a stop codon to form somewhere in the mRNA. A frame shift mutation are insertions or deletions of bases in DNA. These mutations interfere with translation and shift the reading frame. Here's an example of a silent mutation. Silent mutations have no effect on the amino acid that they produce. They are often found in the non-coding region and don't impact which amino acid is incorporated into a protein. So if you look at the top strand, of the wild-type DNA template strand and read from the 5 to 3 prime, you'll see that a mutation is introduced. A silent mutation is where an A is there instead of a G. This still produces the same amino acid sequence. So because there's a mutation that doesn't impact the amino acid which is incorporated, this is a silent mutation. Here's an example of a missense mutation. Let's look at the wild-type DNA template strand. In this case, you have a T instead of a C in the last codon set. The initial amino acid sequence is a methionine, a lysine, a phenylalanine, and a glycine. With the missense mutation, you have a T instead of a C. So at the very end, instead of a glycine, you have a serine. This substitution for one amino acid for a different amino acid can impact the primary structure. So now the polypeptide chain is different than the wild-type strand. This impact on the primary structure of the amino acid can also impact whether or not this amino acid polypeptide chain can form proper secondary, tertiary, or quaternary structures. It can also impact whether or not this protein will function properly. A nonsense mutation is given as an example on this slide. A nonsense mutation involves a base substitution that causes a premature stop codon in the mRNA. The example given here is that there is an A instead of a T in the template strand, and because of this, there's a premature stop right after the methionine. This obviously impacts the primary form, and it will definitely impact the secondary, tertiary, and quaternary structures of this protein, as well as its overall ability to function properly. Finally, we're going to look at frameshift mutations. Frameshift mutations can be a result of either an insertion or a deletion. These insertions or deletions can result in mutations that change amino acids and can result in silent mutations or premature stop codons. Frameshifts alter how the mRNA is read and therefore can alter all the varying structures and functions of the protein that result from the mutation. We've talked about the fact that these mutations can occur in two different types of cells: the somatic cells and the germline or the sex cells. The somatic cells are not the egg and the sperm precursors. They are specific to various sections of the body. So somatic mutations occur in non-germline tissue and therefore cannot be inherited. Germline mutations occur in stem cells that either form egg or sperm, and because of this, these germline mutations can be heritable. So in this lecture, we have covered translation, including post-translational modifications, as well as how DNA mutations can impact translation. If you have any questions about this material or the assigned reading for this material, feel free to email your professor, to come to office hours, or to bring your questions to class. Additionally, you can reach out to your learning assistant who is assigned to this class and who is eager to support your learning in this course. Finally, now that this lecture is over, make sure that you've combined these notes with the notes that you took while you read the assigned material. Once you've combined those notes and reviewed it, use the assisted reading questions as well as the questions at the end of the chapter and at the end of each chapter section as a way to review what you know from this material or where you may have additional questions. You can also use the assisted reading questions and the chapter questions as a way to review and prepare for the quiz and the test on this material.